everyone. My name is Justine O'Brien, and I'm here today to give a short presentation on my secondment that I'm carrying out over in Experimentica in Finland at the moment. So as part of my Crystal Cube secondment, I'm coming from the University College Dublin in Ireland, where my supervisor is Professor Brendan Kennedy, and the host institution, as I said, that I'm attending is Experimentica, which is a non-academic institution over in Finland, where I'm being supervised by Giedris and Mark. The duration of my secondment is 1.27 months, which is approximately 5 weeks. And I started here in the 14th of February 2022. So my secondment is currently ongoing at the moment. So I'm just going to give a brief overview of what I hope to achieve while on this secondment with the Crystal Cube. So a little bit about myself then. So I'm from Dublin in Ireland, and I work as a pharmacologist in the ocular pharmacology and genetics group in University College Dublin, where my supervisor is Professor Brendan Kennedy, and my research aims to find compounds that restore vision in zebrafish models of blindness. And as part of this research, I'm hoping to submit to get a research master's thesis. My research interests include pharmacology, drug discovery and development, compound screening, and also in vitro behavioral assays. So the objectives of my secondment fall under the work packages 1 and 2 of the grant. So the overall objective of the secondment is to understand the safety and tolerability of these 2 compounds known as A2 and 3E in mice models, and the capacity of these compounds to modulate cystine pathways and receptors. So both of these compounds actually were previously found to restore vision in zebrafish models of blindness, of inherited retinal degeneration in particular. So we're hoping that we'll able to see that they're well tolerated in mice, and then to understand how they're modulating the cystine receptors and pathways. So this falls under the 2 work packages, which are work package 1, which is reporting cystine significant in ocular disease, and also work package 2, which is reporting on the therapeutic efficacy of cystine modulators. So the tasks that I will be carrying out include things such as intravitreal training, slit light exam and OCT data analysis, postmortem tissue collection, and functional ERG training, and also I will gain industry exposure during this secondment. So this is a brief overview of the study protocol. So this is the part of the secondment that will be carried out in Experimentica. So we will have 7 different groups. So the first group being vehicle, and then the other groups being both 3E treated or A2 treated groups, each having increasing doses of the compound. So starting with dose 1 being the lowest and dose 3 being the highest chosen dose. So what happened on day 3 is that we will perform the optical examination using a slit lamp. We'll also perform the OCTs to look at the retina, and then intravitreal injection into the eye to deliver the compounds. And these 3 techniques will also be performed on day 3 and day 6, which is our final day. And on the final day, we'll also take samples. So one of the eyes will be taken and will be frozen for protein analysis or RT-qPCR, and the other eye is going to be used for histology. So as I said before, we're taking these mice tissue samples, so these eye samples, and we're sending them then back to UCD, where we'll perform the analysis to look at the cystine expression. So this will either take the form of Western blotting and looking at protein analysis, or RT-qPCR, where we'll look at gene expression analysis to see how these compounds are modulating the cystine pathways and receptors, and also just to see their significance in ocular disease and in the eye in general. And then as part of the kind of secondment impact, obviously in terms of the project, we're going to understand the capacity of these compounds to modulate the cystine receptors and the potential use of these compounds in general in treating ocular disorders. And my host institute, so Experimentica, is going to gain understanding of the usage of zebrafish screening and as a model for ocular disease. And I've already given a presentation to them on this and on my research. The sending institute, so UCD, will gain skills on the use of rodent ocular models. So I'm being trained on all these techniques while I'm over here, so such as the optical examination and also the OCT. And then in terms of career development for myself, they'll get data for publication of my research master's thesis, also hopefully preparation of a paper as well. And I've gotten training in new in vivo ocular techniques, and as well as kind of networking experience and general work experience in an industry in a contract research organization. So that's it for my presentation, and thank you very much for listening.